morning there, Chair Committee and also colleagues. Let me introduce my project and also progress today. So my thesis title is The Role of microRNAs in Renal Fibrosis and Their Molecular Pathways. So I'm a third-year PhD student of the Institute of Translational Medicine. So my vision is to help the patients through translation and basic medicine. So during my PhD degree, what I'm trying to achieve is to find out the novel microRNA that play an important role in kidney disease. So I do have two specific projects regarding this all theoretical background. The first project is basically focused on microRNA profiling studies. The second one is focused on diagnostic accuracy of microRNAs in chronic kidney disease. Let me start introducing the first project. As I mentioned earlier, the microRNA, in this project, we are trying to find out which microRNA is the most dysregulated microRNA, specifically in early and late stage of chronic kidney disease. It would be in systematic and meta-analysis. So at the beginning, I would like to set the stage by introducing background. So I would say the chronic kidney disease is a silent killer, like the 10% of the world population is affected by kidney disease, but those people can lose up to 90% of their kidney function without experiencing any severe symptoms. So that will increase the awareness for search for new biomarkers. Probably that could be in microRNA. So in the last 20 years, microRNAs got attention, so it has the potential to target the gene and also affect the protein expression in any kind of disease. So at the beginning, we were quite wondered about what kind of systematic and meta-analysis could be done. But if you look at the scheme, microRNA has a lot of interaction between protein, also drug, and also molecular pathways, even for any kind of disease. So we try to consider all this interaction in our meta-analysis. So we focused on target gene and also molecular pathways. Our main clinical question was, which were the most dysregulated microRNAs in human and also animal? So our PE was humans and animals tested for microRNA profiling, which means not only single microRNA, for example, like bunch of microRNAs, thousands and 100 microRNAs. And exposure was presence of CKD in both animal models and as a human. Comparator was only healthy individuals and also animal model. So our outcome is most dysregulated microRNAs by ranking order. So let me introduce the methodology because we used like several step-by-step methodologies. So at the beginning, we had thousands of articles, of course, and then we were interested in microRNA array and also sequencing and also some PCR, which has like more than 96 well played. And then we did the systematic search. Finally, we ended up like human 32 and animal 13 study. The main analysis was robust aggregation, as basically in order to do this analysis, we have to rank all the microRNAs by one by upregulate or downregulate direction. And then we used our language to do the analysis. And afterwards, target gene determination, also enrichment analysis using bioinformatic tools. So let me go to the results. So our main analysis was robust aggregation. So outcome will be up and down regulated microRNAs. We have like 3 different subgroups, human results or animal, or different kidney disease by etiology, or different sample types by, for example, urine, blood, or tissue. Also, if you look at the scheme, like there's 3 different pools for human based on the sample types. For animal studies, specifically, it was actually mice. Only one pool we used based on the kidney tissue. So now I'm going to introduce the classical example of our results. So in the x-axis, you can see all results one by one by each author. In the y-axis, you can see the significance of the dysregulated microRNAs. For example, this is the example of robust aggregation from mice kidney tissue and also upregulated microRNAs. So the first 6 microRNAs are most significantly upregulated in mice model of kidney tissue. So you can basically explain the results in this way. So we had like a bunch of this kind of graphs based on all study pools and disease and also biological sample types. So for the interest of time, I would just jump to the summary results. So for On the right side of the screen, you can see the human results. In a urine sample, we found 4 downregulated, 1 up. In a blood sample, only 2 and 1 up. In kidney tissue, there were only downregulated microRNA. For mice model of kidney disease, we found in 2 downregulated, and in diabetic model kidney disease and UEO, basically it's in chronic kidney disease model in mice, we found several upregulated microRNAs and only 1 downregulated microRNAs. So from these microRNA profiling results, the only 2 microRNAs was not validated by PCR in any kind of literatures, and so we are working on these 2 microRNA, determining in FSGS and diabetic nephropathy patient and also kidney proximal tubular cell line to validate this data. So since we investigated the dysregulated micron as chronic kidney disease, our next question was, what is the dysregulated micron early or in a late stage? Because the diagnosing patient in early stage is most important kidney disease. So interestingly, in early stage, there were only few microns were upregulated. In a late stage, tons of microns were downregulated. So this is quite interesting graphs. You can see the target genes by different urine samples. The target genes actually varied by different microRNAs, like 7,000 to 100. We use these target genes for molecular pathways. This is the one example of pathway enrichment analysis from mice model of kidney disease. So most influenced pathways were like fatty acid, prime disease, ECM receptor interaction, and the steroid biosynthesis pathway were most involved in kidney pathology and pathological process. So in conclusion, through the systematic and meta-analysis, we try to conclude the sample and also stage-specific microRNA signatures in chronic kidney disease model, and also we try to interpret the translation of marine model of CKD in human side, and we determined the molecular pathways of microRNA, which was mostly fatty acid pathway. So we can also highlight the study implication analysis strength and limit in detail. So for the submission, we submitted these articles for scientific reports. We got 2 rejection previously from different articles, so it's submitted status. So now I will jump to the second project. For this project, we were interested in diagnostic accuracy of microRNAs also in chronic kidney disease. It's a systematic review and meta-analysis. So for the introduction, as we all know, there is like conventional biomarkers, for example, eGFR and proteinuria in clinical settings, but we cannot really deal with the kidney patients until now. So there is a golden standard test, kidney biopsy, but it's invasive, and so we cannot really do the kidney biopsy in early stage of kidney disease. So we asked maybe a microRNA could be sensitive biomarkers in the future. Our main clinical question was, what is the diagnostic accuracy of microRNAs in humans diagnosed with chronic kidney disease compared with healthy individuals? So we set the PIRD framework, and the kidney diagnosis has to be proven by biopsy or clinical parameters at the hospital. And we did the systematic search according to our search key. And now for the interest of time, I will jump to the results. So the main primary outcome was pooled sensitivity and specificity. As you can see, this RSC curve, and this is the results from only single microRNAs. So the blue dots will show you the sensitivity specificity for diabetic type 2 diabetes patients compared to diabetic nephropathy patients. So in a summary, we can see like the comparison between healthy controls was pretty much the high diagnostic accuracy. It was like 91 and 89% in diabetic patients. We also had an interest in lupus nephritis patients in compared to systematic lupus erythematosus and also healthy controls, in which also the comparison between SLE and also result sensitivity was a bit higher. For example, like 84%. Sensitivity specificity was 81%. And this is the results from panel microRNAs. In panel microRNAs, we only had a chance to do meta-analysis for diabetic nephropathy patients. And there is the diagnostic accuracy of panel microRNAs like 89 and also 73, as you can see here. And then we also did analysis for pooled AUC values, which will show you the overall diagnostic accuracy for specific microRNAs. This slide will show you the summary of panel microRNAs. So we only had us a chance to pull the data on diabetic nephropathy. It was pulled, the results was 86. And the status of this project, we already finished the manuscript, so it's under review within the team. We were planning to submit a second project in July for next week. So thank you very much for your attention.
Thank you. So based on all of these results and everything you know about this topic, what is your opinion about the relationship of the change microRNA pattern and the disease itself? So the changed pattern is the very basic reason or a consequence? Or could be both of them, and or could be changed during the course of the disease? What is your opinion?
Thank you also for the question. I think during also this research, I found there was like one cohort study that tested microRNA kidney disease, and it was sensitive to detect the kidney failure in kidney transplant patients. It was better than the proteinuria and any other like already set biomarkers. I think it could be the one, the biomarkers that we can check the causality, not like afterwards, maybe in early biomarkers. And also from my personal observation, we also did the molecular pathway analysis, not just see like which micron is up and down regulated. We also try to understand what's really happening behind this pathological process, and it was a fatty acid molecular pathways. But interestingly, and also probably, it was pretty much to match the other results that's published from gene sequencing, any other results, studies, not only for microRNA. In this regard, maybe I hope this dysregulated microRNA can predict kidney damage, even from urine samples and blood sample, not only required to have kidney biopsy.
Thank you very much. Please allow me to stay sitting. As I understood from your tables, you included in your study also very, very rare kidney diseases like IgA nephropathy. Am I right? And as I understood, you addressed with this study mainly a functional dysregulation with these dysregulated microRNAs. Have you analyzed, have you compared these increased or decreased signatures with different stages characterized by low eGFR? Stages of CKD, I mean?
Thank you very much. So when we collect the data, it was quite hard to determine whether the patient was an early stage or late stage, but we used the eGFR to determine the patients in early and late stage. And so we had two groups, early and late stage. And after we get the summary results, we try to have two separate analyses in early and late stage. And so early stage, there were only few microns were upregulated. But the most of the data came from late stage kidney disease because the studies were included late stage kidney disease. And so regarding the specific kidney disease, we also have a subgroup mini analysis by each kidney disease, because it really doesn't make sense to combine all different kidney disease at the same pool. And so, but the study, the raw data from each articles was low, so it was really complicated to do meta-analysis based on this data. And so we also have interest for specific disease, we also have results, but it's not ready to publish or submit in any kind of journal. So
thank you. And just one more short question. Is it okay? Did you use cystatin C measurements for calculating GFR? Cystatin C, I mean, instead of creatinine.
Thank you. Only few articles, 2 or 3 articles, had data on C-cystatin. In this regard, we cannot really have a conclusion based on the cystatin.
Thank you very much.
I have a question, what is somewhat related to my question of my colleague. So what do you think, what can be the clinical impact of these microRNAs as diagnostic markers? So I mean, can you diagnose the disease in an earlier stage, or can you subspecify the disease better? Can you start the treatment earlier? Can you make a more accurate, more specific treatment to different stages or in the different stages of the disease?
Thank you for the question. That's actually what we are willing to bring up the science and then show them like, hey, these microRNAs are dysregulated, so we have to consider these microRNAs specifically. And there's a few systematic and meta-analysis. If we publish the data, it's going to be also add some contribution. But also, I think based on all the literature, I think it's possible to diagnose the kidney damage. But it should be narrowed down by specific cells, for example, like proximal tubular specific or blood specific, because microRNA also differs. Circulating microRNA, also the microRNA is only expressed in tissue. So we also have to consider all this aspect, but it's possible, from my opinion.
Thank you.
So yes, what I'm interested in your second study, you show us a very nice ROC curve from the sensitivity and specificity of your result, and what we see very not often, but sometimes we see another pair, so the positive predictive value and the negative predictive value. Why it's good for us to show it, and why you are not reported us this?
We didn't pull the positive predictive value or negative predictive value from each study. Basically, we only pull the sensitivity, specificity, and also pull AUC values. But there's a few articles that provided the data. It's possible to do analyze based on this PPV and PV. But it's, of course, it's a clinical significance if we can predict the positive predictive value specifically for this kind of chronic kidney disease. That will more beneficial in clinical settings. Basically, this results, why didn't we do analyze for PPV and PV, is because of the raw data and availability of the data. So we don't have any results on it yet.
Yes, thank you very much. I'm asking it, for example, because in this situation, I think the prevalence is maybe similar for everywhere, so it will be easier to use the PPV and then PV.
Yeah, thank you for your comments. I will consider this with my statistician.
And if I can, I ask another one. So you showed in your first study as about the animal models.
Yep.
And what do you think, how good this model? So is it a good idea to create additional studies with animal models because they are very useful and not needed patient? Or you think it's meaningless because it's not really working and make prediction for the humans?
Usually we use the animal model if we cannot access to the patient directly, specifically for this kind of gene studies, microRNA testing, and also. So I think animal model will help us to understand the human disease in laboratory model. So if we can collect the samples for animal, we can basically inhibit or increase the expression of this microRNA and see or prove in a laboratory like whether this microRNAs really play in a role in kidney disease. In this regard, it's better to compare animal model with the human data and see whether it's really matching with the human side or not. If it's totally different, we should consider to focus on human samples or in human data.